everyone, welcome back to Cosmic EduTech. I'm Dr. Priyanka and today we are going to decode one of the most revolutionary techniques in biotechnology, PCR or polymerase chain reaction. This technique is very crucial for NEET 12th class students then life science competitive exams. Watch till the end for some memory hack to never forget PCR in your exams. PCR or polymerase chain reaction is a technique used to amplify a specific segment of DNA which will lead to creation of millions or billions of copies of small initial sample. Imagine you have just a very tiny sample or the amount of DNA and you want to analyze it, clone it, to perform some study, for example, to detect disease causing genes. So what technique you should go for? So PCR is the answer because PCR is going to help you create enough of DNA material for analysis within few hours. So this particular technique was developed by Carey in the year 1983 and he was awarded the Nobel Prize for the same. So this technique led to the production of millions of copies of tiny sample of DNA. Now let's see what is the principle behind PCR. The principle of PCR is based on the natural phenomenon that is the replication, DNA replication. So we all know that DNA replication occurs in the cells. The same procedure is being conducted or is being worked upon in vitro, that is in some tube that is outside the living organism. So this technique was developed by Carey. So here there are certain different things that come into picture and the most important one is the enzyme Taq polymerase. Whereas in case of your actual DNA replication, we know that DNA polymerase is the main enzyme. But in case of PCR, there is yet another enzyme which functions almost similar like that of DNA polymerase is the Taq polymerase. Now this Taq polymerase is heat stable, okay, and it synthesizes new strands of DNA using the existing strands as the template. This polymerase was developed or isolated from a bacterium called as Thermus aquaticus, which is found in the hot springs, allowing them to withstand the high temperature. And this is the very point that has been picked up by Carey and he has used high temperatures in case of PCR. Let us see what are the requirements of PCR. First of all, we know we should have a template DNA which will be amplified further. Then we have the primers, the short single-stranded sequences that define the region that needs to be amplified. Next is the DNTPs, which are the free nucleotides. Taq polymerase, the main enzyme, so enzyme that is going to synthesize the new strands. And finally, the buffer solution, which is going to maintain the optimal pH and the ionic conditions. So all these are mixed in a PCR tube and it is placed in a thermal cycler, the machine which is used for PCR. And here this machine is going to make precise changes in the temperature and it is going to lead to the rotation of the cycles. So next thing, let's talk about the steps which are involved in PCR. Now PCR is mainly comprised of three steps, that is the denaturation, annealing and extension or elongation. These are the three steps which are involved in PCR. So now let us see what happens at each step separately. So first let's begin with the step that is denaturation. So two things to be remembered over here, that denaturation occurs at higher temperature, that is around 94 to 96 degrees Celsius. So what do you mean by denaturation? Denaturation is the separation. It is the separation of the DNA, double-stranded DNA. This occurs by breakdown of the hydrogen bonds. The increased temperature is going to facilitate the breakdown of the hydrogen bonds between the single strands of DNA. And as a result, we will have two templates here. Think of something like, unzipping of a zipper. So that is what happens at the denaturation step. Point to be remembered is the temperature range, that is from 94 to 96 degrees Celsius. Let's talk about the next step, that is the annealing. Annealing is something like binding or joining. So here the temperature is now slightly lowered from the previous stage. That is, here it is somewhere around 50 to 65 degrees Celsius. The lower temperatures, they are going to allow the primers to bind or anneal to their complementary sequences on the single-stranded DNA. And these primers, they act as the bookmarks which will define the point from where the copying of the DNA has to start. So here the point to remember is there is the annealing temperature and involvement of the primers. So let's move on to the next step, that is the extension or elongation. So this is the next stage of the PCR process. So here exactly what happens in case of extension or elongation. Now here again the temperature is slightly increased to somewhere around 72 degrees Celsius. Now Taq polymerase is going to add free nucleotides, that is DNTPs, to the 3' end of the primers, and hence synthesizing new DNA strand which is complementary to the template. So these steps together, they are going to constitute one cycle of PCR. So if you see this image, okay, here at the 3' end, you will see there is the continuous addition of the DNTPs leading to the extension or the growing of the strand. Generally, there are around 25 to 35 cycles are being run, which will result in exponential amplification. For example, one copy of DNA can give rise to somewhere around 1 billion copies after 30 cycle, and this is called as amplification chain reaction. So let us move on to the next part and see what are the applications of PCR. PCR has vast applications in science, medicine, forensic science, okay, research areas. So medical diagnostics, that is detection of pathogens like viruses. For example, in case of COVID, there were, RT-PCR tests being conducted, and then it can also look for the, enhancement or amplification of the genes involved in the infections. Genetic disease detection, so identifying the mutations in the genes. For example, in diseases like cystic fibrosis or sickle cell anemia, these are the genetic diseases which can be detected using PCR studies. Then forensic studies, DNA fingerprinting at the crime site in the crime investigation and legal identification. Next is molecular cloning, amplifying the genes for cloning and recombinant DNA technology and studies. Paternity testing, so you must have seen this thing in most of the Hindi serials or whatever serials. People talk about claiming the paternity. They say we are waiting for the result. Actually, they are not waiting for any other result, but it's the PCR test. So here they are going to compare the samples, DNA samples, and establish the biological relationship between two people or n number of people. Then lastly, research applications, gene expression studies, sequencing, evolutionary studies. So these are a variety of areas which can be explored using PCR. So actually, if we go to say without PCR, modern genetics, diagnostics will be very, very impossible to go about. Finally, let's talk about some easy way to remember PCR. The steps in PCR, we can remember DAE, that is denaturation, annealing, and extension. So, and another thing that we have to remember is the play of words here, that is Taq. Taq polymerase is obtained from Thermus aquaticus. So it is rearrangement of these letters here in Thermus aquaticus. So these are some of the ways you can remember the steps in PCR. I hope PCR is now crystal clear for you. And if you like this explanation, please like the video, share it with your friends and study groups, and subscribe to Cosmic EduTech by Dr. Priyanka. So for more such videos where we are going to make complex topics simple, comment below for your next toughest topic that you want to make more simple. So till then, keep smiling and keep studying.